Okay, do you want?
Sure.
I have pockets in these pants.
Perfect. All right, everyone. Before we get started with the lecture, I just wanted to make two quick announcements. The first one is we're still finalizing a couple of office hours availabilities, but there are still we still have like 3 out of our 5 office hours finalized that you guys that are available to you. I only had one person come to mind, which like isn't an insult towards I don't personally, but rather it's I just want to articulate that the office hours is made available for you guys, and it's a really important resource, I think, through the material. And I've seen huge improvements. This is my 4th time teaching this class. It's pretty remarkable to see students' grades go up the more they attend office hours. So I highly, highly, highly encourage you guys to come to office hours. We have a teaching team, and we're working to make sure that office hours are as accessible as possible. Once we have them finalized, I actually might send out a quick email to the class to ask, do these office hours work for everyone's schedules? Because I know we have, historically, this was like primarily like only like second years in the class, but now we have like a vast diversity of students across years in the course, which means schedules aren't as standardized, and it can be hard to predict what times are available for everyone or what works for everyone. But I want to just really articulate, I highly encourage you guys to come to office hours. Maybe it's better if I call it student hours to kind of identify that like these are dedicated times for you guys for us to be available to you guys, for you guys to come. Maybe I'll bring treats. That'll help. But yeah, so I highly, highly encourage you guys to go to office hours. It's a great resource. It has measurable changes in students' grades in terms of how it helps students really digest the material. I think the last 2 years I had like a cohort of like 8 to 10 students that would always come. I'm pretty sure everyone either got like an A or A plus and come to office hours. I'm seeing nodding, so I'll take that as an affirmation. But yeah, so like it's and that's not like me handing out the grades, but rather like it having a meaningful impact in understanding the material and working through it. The second announcement is for the projects. So next week, the first assignment is going to be due. All that assignment's going to be is you identifying what the disease or biological problem is going to be that your project is going to be focused on. It's going to be just like a really brief paragraph. Describing what that disease or biological problem is, and to help inform you as you make your decision. It can absolutely be something that could either be something you're very interested in or passionate about, something that you have a personal connection to. We want to make sure that it's going to be a cellular or molecular biology, something that has a cellular molecular biology element to it. And the idea is that as we work through this project, you guys will be going through the literature to understand what's known and what's unknown about this disease or biological problem. And particularly, you're going to be proposing either a mechanism, if there's an unknown mechanism for this disease or biological problem, or an alternative therapy that's going to be better than what's already available. And so those two key pieces that are going to, I think, might help guide you as you try to identify what your disease or biological problem will be for the semester. And I should also note that I think our both Najwa and Mackenzie can agree with this. In science, as you're understanding things and as you're working your project, things can shift or things can pivot, and that's okay. Like that's literally how science works. As you understand things, you might alter your approach. So I don't want you to be worried that when you submit your assignment, you are absolutely locked in and your fate is sealed. But in reality, like science, like as we learn new things, things can pivot a little bit, things can change, your approach might change. So the idea is that we're doing these assignments throughout the course of the semester so that you can stay on track and not have to work on all of this the night before it's due at the end of the semester. And so are there any questions? Oh, and I should also say before I forget, office hours are also to come and get feedback on your project. If you want to get a sense of if your biological problem or disease will be a good thing to focus on for the semester. Are there any questions on either office hours, projects, or particularly the assignment that's due next Tuesday? Yes. That's a great question. So you'll be working on this, what you submit, you'll be working on individually, but you'll be getting feedback from peers, each element of the project, meaning like the 3-minute thesis, the graphical abstract, and the 2-page research statement. And we'll be trying best to try and almost put you guys in study sections where people who are working maybe on like neurological things or maybe on tissue engineering or any cardiovascular disease or cancer, like anything, like we'll be trying to like orient you guys in like similar groups so that you guys can have so that what you're learning about your project might help inform others, but then also your strengths and weaknesses might lift each other up. So it's going to be, you'll be working on it individually, but getting feedback, getting peer feedback, and giving feedback as well. That's a great question. Are there any other questions? I love questions. I love dumb questions because then it's easy for me to answer. So I also invite, yes. Cellular or molecular biology, yeah, that's the idea. And so like, perhaps perhaps the disease that you're focusing on can have, might not be, there could be different elements related to that disease, but the therapy that you're going to be proposing should be a cellular molecular biology focus. Yes. Yeah, I would say start with the disease and then look at the therapies, because I would say it's possible that there are no currently available therapies. There might not be a known mechanism. So I would say start with the disease, read about it, dive into literature, trying to understand what are the gold standard therapies, if there are any, and then based on what's known and based on what you've been learning, try and start working towards proposing an alternative therapy. These are all great questions. Yes. The submission will be on Canvas, and so we'll go. This is actually a helpful reminder. So we'll actually go ahead and post the assignment on Canvas, and so the deadline will be Tuesday at like 11:59 at night or something like that. Yeah. Great question. Are there any other questions? All right. So this is not the last opportunity to ask questions. If you have any other questions about either office hours or the project, you can come up afterwards, or you can also come to office hours. Another plug. Or you can send us an email. Thanks, you guys.
And just another quick announcement, the grades for your first quiz will be posted today. So they're all graded right away. Just had to input them and it's just hidden right now. So we'll make that available today so that you guys can just keep track of your grades and make that decision if you want to keep focusing on the quizzes or the exams or both. But we can go ahead and get started. So for the lecture today, we're going to be talking about the extracellular matrix. And our disease topic that we'll use to demonstrate how this key component of cellular biology plays out is pulmonary fibrosis. So a couple of the learning objectives today, we'll review some of the structural fibers in a little further detail from what was covered in the pre-lecture. We will talk about the hydration component of the matrix, as well as different cell adhesion molecules that are linking cells to cells and cells to the matrix. And then we'll finish with the overall theory of how pulmonary fibrosis progresses. I forgot to delete in COVID-19, but COVID-19 is a big risk factor for pulmonary fibrosis. So this is a disease that is of great interest to me as well as Dr. Ababayu. We've both studied pulmonary fibrosis in depth, and what this means. So pulmonary just refers to your lungs, and fibrosis is scarring. So this is scarring of the lungs, and in particular, this scarring does not resolve. So it increases over time, and of course, this is an issue in your lungs and occurs in many organs. There's many different manifestations of fibrosis, but in the lungs in particular, it's happening in space where oxygen exchange is happening. So this prevents oxygen to be transferred to the blood, and ultimately makes it harder and harder to breathe as that scar tissue progresses. So some obvious symptoms that might be caused from this is shortness of breath, a persistent cough, fatigue as your tissues don't get enough oxygen, and weight loss. And one aspect of this disease that makes it particularly compelling to study is that there is no current cure for pulmonary fibrosis. So different diagnostic criteria are used. There's different subtypes of pulmonary fibrosis, but the ultimate treatment is just palliative care. So oxygen, there's different therapies that can help to slow the progression, but nothing to stop or reverse it. Some patients are eligible for lung transplants, but interestingly, sometimes pulmonary fibrosis can redevelop even after a lung transplant. So it's a really interesting disease with a super poor prognosis, about 2 to 3 years from diagnosis, which is just a term for how long patients can live after they're diagnosed with the disease. So causes of pulmonary fibrosis. The most commonly diagnosed form of pulmonary fibrosis is called idiopathic pulmonary fibrosis, or IPF. And the word idiopathic just means that they don't know what triggers it. So we don't know the etiology of that disease. There's likely many contributing factors in this pathology, but it is thought to result from abnormal tissue repair. So we'll talk about this at the end of the lecture, the difference between normal tissue repair and fibrosis. But there are a couple identified risk factors. So exposure to different aggravants to your lungs, so silica dust, asbestos, are both two key ones that have caused many different cases of pulmonary fibrosis. Mutated TERT, so short telomeres, this is something that happens with aging. So age is a very primary risk factor for pulmonary fibrosis. And recently, COVID-19 has been a big trigger for pulmonary fibrosis. Just another one of these cues that are an aggravant to your lungs that might be causing some type of tissue damage. So in terms of the prevalence and incidence, there's about 20 people per 10,000 worldwide that are diagnosed with this, which is a lot. There's actually as many people affected with pulmonary fibrosis as breast cancer, and 100,000 of those people in the US currently. So this is definitely a compelling disease to study. I think my PhD is about. So what this looks like in clinical samples, so this is a slice of the lung that is stained with things that can identify cell nucleus and cytoplasm. So the areas of dark pink are where there's a lot of cells, and this is the lung, of course, so there shouldn't be a lot of dense packing of cells. There should be a lot of air space available. And some of the things pulmonary fibrosis is a lot of heterogeneity. So there's honeycombing is one way that is particularly useful for diagnosing the idiopathic version. This is a key trait of IPF in particular. Not you see kind of in this area. Oh, I don't know my laser. In this area, so the pattern of scarring looks kind of like honeycombs, but there's where it's really dark pink, and then there's all sorts of other, so there's normal interstitium. We'll talk about what interstitium means, and then the fibroblastic foci area of active scar progression. So there's a lot going on in this disease. It's very dynamic. There's still a lot to understand. And we'll just touch briefly, about the mechanism that we know. Question? So there's radiation-induced pulmonary fibrosis. So sometimes if people get lung cancer, the exposure to radiation causes lung fibrosis. That's definitely a common one that's diagnosed in the clinic. For things with known risk factors, that wouldn't be idiopathic. So if COVID-19 triggered your pulmonary fibrosis, it would be COVID-19 induced pulmonary fibrosis. Or if you worked in a silica mine your entire life, and that is a reasonable cause for pulmonary fibrosis, then that would be how the disease is then named. Good question. Thank you. All right. Any other disease? Yeah.
cystic fibrosis before, we both affect the lungs. But what is the difference there? I know one one's fluid and this one's scar tissue, but
Exactly. That's a great question. I'm not sure if they can develop in the same individual. They're definitely, as far as I know about cystic fibrosis, not necessarily overlapping mechanisms. Cystic fibrosis is more of a genetic disease, whereas this is something that there's thought to potentially be a genetic component. That's actually one type of pulmonary fibrosis, familial, if they can link it to a specific gene. But in terms of idiopathic pulmonary fibrosis, there's not a known genetic factor. It's something that is happening to you environmentally. So in terms of the cause of the disease, they're very different. In terms of the actual pathology, the mechanism of the disease, like you said, cystic fibrosis is more of a fluid buildup in your alveoli, which are the air-filled sacs that do the transport of oxygen at the terminal ends of your bronchioles, or just lungs. So yeah, a lot of people ask that question. It's a really well a lot of people know about cystic fibrosis. So the word fibrosis there can throw people off, but cystic fibrosis is less about what we think of in terms of fibrosis, which is the scarring process. Good question. Yeah, there's a lot more understood about what causes it. And this lack of treatment is actually not necessarily specific to pulmonary fibrosis. So kidney fibrosis, liver fibrosis are two other types of fibrotic diseases that present and that also don't. Questions? I'm going to go ahead and progress into the lecture material. So before I get going, was there any questions about the pre-lecture? All right. If you think of any, feel free to speak up. So this slide is a little bit of an overview of the lecture, as well as the different ECM components. So the extracellular matrix, we can think of this as the scaffold around which cells are surrounded. And so they live and survive, and this is not just a structural component that the cells exist in. It's very active and dynamic. There's a lot of signaling that happens between cells and their matrix, and there's all different types of proteins that are interacting with each other in different ways, as well as with the cells. So we talked about a couple of the structural fibers in the pre-lecture. We're not going to talk about collagen in this lecture all that much. We're going to hit on elastin a little bit and then through the rest of these. So I first wanted to start out with what is a hydrogel? You might have mentioned that was a blue word on the last slide. You can think of the extracellular matrix as a hydrogel. So this is just any kind of polymer network that is cross-linked and that is swollen with water or some type of fluid. So we'll talk about the fluid that is giving this hydration to the extracellular matrix in our bodies. But this is exactly the same thing as like what a contact lens is made out of. So these are hydrogels, they're polymers that have water in them to the fluidity of your eyes so that they can transmit light that way. And this is a really clinically. So I bring this up because this is a really important field in bioengineering. There's a ton of clinical applications for hydrogels, and all of this work has really been inspired by the native structure of the extracellular matrix, as well as its function, its mechanics, and other things. So wound healing is a big one. So there's currently a lot of hydrogel collagen-based biomaterials that are used as a dressing for skin wounds. And this has been really important for burn patients and other people that experience really drastic wounds on their skin. Drug or cell delivery, this has been really important. So This can be used for any type of cell-based therapy. A lot of people will put cells in a hydrogel to protect them while they deliver the cells, to keep them alive in an environment that's more similar to their own. Research. So this is a big part of what we do in my lab. We make hydrogels, and this is kind of a progression in terms of how we study cellular biology. So these hydrogels are more similar to the environment that cells exist in the body. And so we can use that to take the results that we find in the lab a little bit further to try to get closer to what cells are actually doing in the body in a way that we can study them in the lab. So we can grow cells in or on hydrogels for research purposes. And there's this whole field known as tissue engineering or regenerative medicine. So this is literally what it sounds like, where you are growing tissues, regenerating new tissues for implantation. And these hydrogels are often used as a scaffold to grow and populate. So you can think of some type of organ. So your heart, for example, you can explant it from one person, you can wash away all the cells, and what would be left is the extracellular matrix, a lot of structural components, such as the collagens that were talked about in the pre-lecture. And then you could repopulate that with somebody else's cells and use that as an implanted heart, for example. So this is a still actively developing field, but it's progressed a ton in the last few decades, and is a really exciting area of work that hydrogels have really made possible. Biosensors is another one. This is a little more complex, but this is a really exciting field where hydrogels can sense something about their environment and then respond based off of the chemistry that's going on. So this is a little further from the native ECM, but can be really useful in a lot of clinical applications. Yes. Mm-hmm.
And one of the applications is published in one of you guys, which is glucose monitoring for diabetic patients. The scaffold of like a hydrogel can be used for cells, but it also can be used for proteins. Like glucose oxidase enzymes within those hydrogels, planted on a sensor, and then as the glucose or whatever it is that you're looking for is oxidized, the electrons are registered at that sensor surface, and then you can monitor it.
Yeah.
So it's really cool, kind of the variety of what you can.
Absolutely. Thanks for adding that, Najwa. The diabetes in particular is a really great disease application for biosensors, since the clinical standard still needs a lot of improvement and is a big disease burden, especially in the US, but hydrogels are a really useful tool. Okay, so a little bit back into the structural components of the ECM. This slide is from the pre-lecture. Elastin is one of these components that make the structure of our ECM. It's amorphous, it's secreted in a soluble form known as tropoelastin, and then it's cross-linked by lysyl oxidase, so this enzyme that was also mentioned in the pre-lecture, and this gives it some rigidity, but ultimately it's balled up in its normal state and can be stretched and then returned back to this balled up state. So it gives this elastic component to the extracellular. So a little bit more about this spring-like behavior. So if you think of a normal spring, if you pull on it, you're adding some potential energy into that spring. And this is working in a little bit of a way. So this is driven mostly through Gibbs free energy. We can think about it through Gibbs free energy. And this is a concept that will be important in a lot of things. I know we've discussed entropy and passing in a couple different lectures so far, but this is important in terms of what gives elastin its elasticity or its springing-like nature. So if we remember the Gibbs free energy equation, we have delta G equals delta H minus T delta S, where delta G is the Gibbs at a positive value. That means there's energy that needs to be input to make this change. A negative value means that when this change happens, there's a release of energy. So that's spontaneous, that's favorable, negative delta G. So we have these two components. We have enthalpy and we have temperature. And so enthalpy changes are a function of chemical reactions. But in terms of elastin, there's no chemical reactions that are happening here. So for that, for now, in terms of this change, so really entropy and temperature would be the driving forces. So focusing on entropy, since temperature is relatively stable in our body. Entropy is kind of a complicated idea, but you can think about it in terms of the degree of order or the degree of disorder is probably a better way to think of it, where increasing entropy is increasing disorder. So as we talked about water molecules, the ability to go anywhere and do anything is a higher level of disorder or increased entropy. And so as we have increased entropy, this negative sign makes this a more negative delta G, which is favorable. So we want disorder, and if you look at how these would be folded, in the most extended state, when there's tons of force being applied onto the elastin molecule, it can only really go into one conformation. So all of these crosslinks are stretched to its furthest extent, and it can only exist as its stretched form, versus if there's not any force being applied on the elastin molecule, it's free to turn and move around all of its bonds. It's a very flexible molecule, and this is a high degree of disorder, which is a high level of entropy, making this favorable. Does this make sense? Is there any questions about this? Again, this would be a great concept to come and talk through in office hours. I know the idea of entropy can be really tricky and confusing, but it's fairly intuitive in this case, how these spring back. Yeah. Yeah, so delta G is the Gibbs free energy. So free energy, Gibbs is just a guy, but he gets the G. So the free energy is like the energy available. And because it's a delta, we're talking about some kind of change in states. So if you have a change in state with a positive Gibbs free energy, that means that that change requires the input of that amount of energy. Where if you have a negative delta G, then that change releases this amount of energy. So if you think about what might be more favorable, if you have to input something, that means that it's not as likely to happen spontaneously, versus there is a release in energy, you're going to a more favorable state, and that would be an overall favorable reaction. Does that make sense? Okay. Yes. Yeah, so this is just showing the change in the number of states that something can exist in, and that's one way to think about order or disorder. So an ordered arrangement would have less states that something could exist in, whereas a disordered structure would have a lot of different states. So like a spaghetti noodle that's just in water can be in thousands of different conformations, right? But if you take it out, it's gonna hang, and then that's the only way that it can exist. So it's one state that it could exist in versus many. So there'll be higher entropy in water versus holding it. Does that make sense? Okay. Yeah, that's pretty much what this diagram is representing. You can think about this process in multiple ways, but I just wanted to make sure we could walk through this equation and make sure you guys have a good understanding of entropy, since it's come up a couple times now. All right, we're gonna move on now to the hydrated component of this. So the hydration of the extracellular matrix is both due to the fluid that's surrounding it, as well as the abundance of proteoglycans, which, if you remember from the pre-lecture, are very hydrophilic molecules that like to draw in water. So this maintains all of the natural fluid that is surrounding these matrices. Okay, this is another slide from the pre-lecture, but an important concept that will come back again in this lecture. So these proteoglycans, proteo meaning protein, glycan meaning sugar. It's a combination, a core protein So that's what is all of these glycosamino branching off, and we can refer to these as GAGs or GAGs. And these glycans are repeating disaccharides. So that means saccharide is a sugar molecule. There's disaccharide, means there's two of them. And so there's these repeating units of two disaccharides. And these disaccharides have this amine modification. So you can see the nitrogen on each of these on our chondroitin sulfate, keratin, and hyaluronate. And they're often sulfated as well. So there's a lot of hydroxyl groups available that can have sulfation of this SO3 group, and that adds a lot of negative charges to these. So these proteoglycans and the glycosaminoglycans in particular are always negatively charged. So they're very polar, attract a lot of water. This is key for the hydration of the matrix. Is there any questions about this? I know it's just a bunch of terminology, but once you're familiar with it, it's good. We're going to be talking about more glycosaminoglycans that are present in the extracellular matrix. But first, we'll talk a little bit about the fluid. So the extracellular fluid that is hydrating this matrix can also be referred to as the interstitial fluid. And what this just means is any fluid that's not like in your veins or in your cells. So outside of your cells, outside of your veins, in the ECM is that interstitial layer. And the fluid that exists in there is perfect for what cells need to survive. So they like to live in this, perfect for ion transport, and the concentrations of different proteins and ions are essential for that. So this is showing the difference between the concentration of various components in your interstitial fluid versus your plasma, which is the fluid that makes up your blood vessels. And you can see that it's fairly similar. Some things that you can note here, there's a high level of sodium as well as chloride. So the interstitial fluid in particular, about 93% of the total cations are sodium and 73% of anions are chlorides. This is a large percentage of the different ions in the fluid. Fluid. So it's generally low in proteins, and this is because these need to be transported across the membrane of your vessel walls. This is tonic, which is a specific word for cells that's similar to osmosis, where it's the difference in solute concentrations inside and outside of the cell. But this specifically refers to that can diffuse in and out of the cell. So these are isotonic, but just about isoosmotic as well, if you think about the differences of the fluids that can, or the solutes that can diffuse in and out. But you don't have to know those definitions for this. It's just important to know what different components make up the interstitial fluid and how it's similar or different to in the clinic, especially for replenishing when somebody is dehydrated, if they undergo a lot of blood loss or vomiting, and the imbalance of fluids can be really harmful. So we have IV fluids that we can use to replenish this, and this is based off of what our interstitial fluids are. The main categories of this are saline, as well as Ringer's lactate, and saline is what is most commonly administered. And Ringer's lactate is a little more similar to the interstitial fluid, particularly the incorporation of bicarbonate. So I didn't mention that yet, but bicarbonate is present in the interstitial fluid, and this is a buffer, which means it maintains a fairly neutral pH, even if there's changes in acidity or base. If there's changes in acidic components or basic components, it buffers that. So it maintains generally neutral pH. No, this is really important for protein conformations, since these different side chains can be protonated or deprotonated with small changes in pH. So this bicarbonate is essentially a fluid, and this is present in Ringer's lactate The reason why it's not always administered is it doesn't mix well with certain other fluids, and it doesn't mix well with blood, the administration of blood. So saline is still the most common. Okay, any questions about these two before we move on? All right. We'll talk about a really important extracellular matrix structure known as the basal lamina. So the basal lamina is a very thin sheet of ECM, and this lines both our as well as our endothelial layers, and our epithelial layers are anything that face the outside of our body. So skin is a predominant one that we can think of, but this also refers to the lining of different organs. So your stomach or your kidney all have an epithelial layer of cells that create the structure around the organ. Endothelial layers are anything that face the inside of your body. So the main cell type that we'd think of in this are cells that make up the inside of your blood vessels. So you can think of them as technically facing the inside of your body. And the basal lamina provides a support just on these. So you can see in this figure here, an example of an epithelium, and then the basal lamina will be just under it. And then everything underneath that fills the other space is known as the connective tissue or the interstitium. So when we talked about interstitial fluid, that's fluid in this layer between different epithelium. And so if you look at this electron micrograph, you can see how thin of a layer this is. These are cells. It's much smaller than cells, and we'll talk about the exact scale of this in a couple slides. But a little bit more into the details of the basal lamina, we'll talk about all of the different components of the basal lamina. So this is collagen type 4, laminin, and this specialized sheet of ECM for a long time was just thought as a structural support. It's also a barrier, which of course is providing a physical barrier. But more recently, it's been appreciated to have more consequences for phenotypic regulation. So cells can bind to different parts of it, and that can change the behavior of those cells that are binding it, different cues, the basal lamina. So we'll start with collagen type 4, but aspect of this is this collagen is much less organized than the collagen type 1 that we talked about in pre-lecture. And so this more disordered behavior of collagen type 4 leads to this basal lamina looking a little bit fuzzy on the electron micrograph. So this is just due to the disordered nature of this sheet compared to that make up other components of the extracellular. Okay, so this is one of the components of the basal lamina. It maintains a triple helical structure like collagen type 1. This is generally something that's found in all collagens, but the disruptions of these helices is different between them. So the collagen type 4 has more interruptions in that helical structure than collagen type 1. And this changes the structure or the packing or the flexibility of these molecules. And these are all fairly interrelated. So you think of this kind of similarly to the saturated versus unsaturated fats, where if you have totally saturated fats, they can pack really tightly versus the unsaturated fats have those kinks in them, which decreases the packing. So these would be less packed because of these interruptions in the but it's still a very strong molecule. These 3 proteins wrapped around each other causes a lot, provides a lot of tensile strength, and is the predominant proteins found in the lamina. This collagen type 4 is also specific to the basal lamina. So it's going to be, whereas things like fibronectin, which we'll talk about, is not specific to the basal lamina. Any questions before I move on? Yeah.
Say that it is like less dense or packed. What would that compare to other types of collagen?
Collagen type 1 in particular. So if you remember back to the pre-lecture, collagen type 1 has this triple helical structure, but then those are organized with others, and then those are ultimately into fibrils, and then those fibrils are organized together. So it's a really densely packed hierarchical structure that creates a lot of strength for these collagen fibers. But this is not that way. Thank you for clarifying. All right, next we'll talk about laminin. So laminin, despite this picture, it's a very large protein. Also has multiple components similar to collagen, where these different subunits are kind of wound around each other. This is thought of as like a bridging molecule. So you can see it has different binding domains. So we've got a collagen 4 binding domain. We've got a cell surface receptor binding domain, a heparin binding domain. A lot of components of the basal lamina have these different binding domains. And that helps the structure to remain tight and interconnected. That provides a lot of strength. This is also a protein that's found almost exclusively in the basal lamina. And this partly because of the specificity of these binding sites. So like I said, since collagen type 4 is primarily found in the basal lamina, has these collagen type 4 binding sites, these are often found together. All right, last couple structures. So nitrogen, also known as intactin, is a glycoprotein. So this is different than a proteoglycan. Proteins that have sugar modifications or some glycation attached. This also acts as a bridging molecule in the basal lamina. So it has laminin, similar. So this is a heparin sulfate proteoglycan. Thinking back to the GAGs, this is the proteoglycan that's that we talked about earlier. And this also bridges laminin and collagen 4. Yes. Yeah, so it's just binding one thing at one end and the other thing at the other end. Good question. Oh, heparin sulfate. So that is one of the proteoglycans. Yeah.
One thing I was going to say really quickly, I can remember being really confused hearing glycoprotein and having a hard time trying to tell the difference between the two. Maybe that's a better person. But kind of like a little neat trick, at least that I learned to try and distinguish between the two, is typically what's on the end is what is like the most abundant thing there. So if like for a glycoprotein, it's mostly a protein with just a few like a small number of sugar molecules added to it. A proteoglycan, it has a core protein on it, but it has a ton of disaccharides of GAGs littered all over it. And so that's like one small thing to kind of tell the difference. And again, like glyco or glycan is just a short-term notation for sugar molecules.
Yeah, that's really helpful. Thanks for adding that. Yes. Bridges just means binds one thing on one end and binds another thing on the other end. So if you literally think of a bridge, it's like a structure that takes you from one piece of land to another piece of land. This is a similar thing with proteins. It's just a protein that links one protein to another protein.
typically like the collagen four name, like you have like two networks that need to be bridged together because like laminate will like self-assemble together and collagen four will like self-assemble to networks, but they need to be bridged together to create like a stable structure. And that's like the key function, like what protein does there. Both protein and additive, they help bridge those networks together to create the base membrane.
Yeah. Great. So the last protein we'll talk about in terms of the basal lamina is fibronectin. And this is a really important protein. This is what the journal club will be all about today. So reminder, we'll be doing that later. But this is not explicitly required for the formation of the basal lamina, but is usually present for binding cells. So this is a protein. It's got multiple domains, and it is generally thought of as binding cells to the matrix. So because the structure of ECM is right next to cells, you can think this is often going to be present binding cells to this basal lamina structure. So fibronectin can exist in both soluble and insoluble forms. So in the matrix, it's insoluble. It's a protein that is assembled, but it's found in insoluble forms in your bloodstream because it's actually going to help. So, these are proteins that are working in your blood clot cascade. So fibronectin is going to be important in cascade goes smoothly. And this contains a lot of other binding sites similarly. So we want this molecule also to be attaching cells to the matrix. So it'll bind different parts of the matrix in different ways. But this RGD sequence is of particular importance. And we'll talk about this more when we talk about integrins in a few slides. So this is a schematic that kind of shows how this all comes together. So I know talking about these components separately doesn't really mean much, but once you see it all assembled, it might make a little more sense. So fibronectin is included in here, but integrins are, which again we'll talk about in a few slides, but are coming from cells. So these are transmembrane proteins. This bottom sheet here is just representing a plasma membrane, so the outside of a cell. And you can see this collagen network and this laminin network kind of interspersed with each other. And then we have our perlican binding them to small in comparison to collagen and laminin. So you might have to squint a little bit to see them. But this structure, all of it is binding together and it maintains a really tight so the sheet can be anywhere from 40 to 120 nanometers thick. In this schematic, it looks really organized and structured well. It's not generally this organized in the body, and it'll build up multiple layers. So this single layer schematic kind of makes it easier to see the interactions, but this can be schematic kind of summarizes those interactions between the molecules that I mentioned on previous slides. This before molecules.
Are the on the cell matrix like significant? Like why is this significant?
Yeah, that's a great question. So the basal lamina is playing this support structure role. So it's right under these different epithelial or endothelial sheets. So if these were just a bunch of interspersed matrix proteins, they wouldn't be able to provide any kind of like structure or support barriers. So because they're all interwoven and bound to each other through these bridging molecules, it can create this really tight structure that forms this barrier between the cells and the interstitial layer. Does that make sense? Yeah. Great, thanks for that question. Yes. The functions of fibronectin?
I saw that it was like highlighted that
the purpose of the anticoagulation cascade. Yeah, so that is just the blood clot formation. So coagulation is just your blood clotting or coming together, coagulation. And fibronectin can bind these different proteins that are causing this. So heparin and fibrin are two primary proteins that make up blood clots. And because of these binding domains, it's able to kind of sequester these all in the site that it needs to. Does that help? That's just one of the functions. The main one we're talking about in this lecture is in reference to the ECM. So binding cells to the matrix. Yes. I don't know what that molecule is. Oh, it's a drug. Huh. It's for what? Sorry. A blood thinner. Huh. Yeah, that's a great question. I don't actually know. If you wanted to post more information on Piazza, I would love to learn or email us either one.
One thing I was going to say, in addition to, I think the question earlier about fibronectin and it binds like anticoagulants. So, you know, one thing we said at the first day of class is that like this is a class. A great way to put it, like this is a class of half-truths, meaning that like things are constantly still being discovered. And so, for example, like one thing that was recently identified is that, so we know that fibronectin binds to like fibrin and heparin, which are parts of the blood clot. And what we've identified is that fibrin binding to like our blood clots is kind of a way of, you know, blood clot is primarily think of it as like hemostasis, like stopping blood loss. But the fact that fibronectin can bind there has been pretty cool because what we identified is that fibrin being there starts to create like a new ECM, like kind of like a temporary new ECM. And that typically we've identified also that the way in which fibronectin is laid down, collagen will like follow suit in the same exact orientation. And so like it's kind of like you can think of fibronectin paving the way for collagen to create like a new, more mature ECM. And then kind of like bring it full circle to the main disease here. That collagen deposition or being laid down needs to be controlled because if it's excessive, you get fibrosis or scar formation.
Yeah, and we'll talk about that in a couple slides. We should get moving because we're running out of time for the general club. I'm so sorry. Adhesion molecules. I really want to emphasize integrins, both because they're important for this paper as well as generally. So you can think of these are heterodimeric proteins. So this just means there's two components. So there's an alpha and a beta subunit. They form this dimer, and the hetero just means that those two subunits are different. So the cells can express different types of integrins with different alpha and beta combinations. And these different alpha beta combinations give the integrin structure some specificity as to what extracellular matrix components they bind. One thing that is conserved among integrins is that they always bind at this RGD. So this is a 3-letter amino acid sequence referring to arginine, glycine, and aspartate. And this 3 amino acid sequence is present in a lot of different ECM components. So this is in fibronectin, this is in collagen, this is in laminin, and many other different extracellular matrix components. And so the function of these integrins is to bind cells to these different components through these specialized structures. So you can think of them as like integrators of the extracellular matrix components to intracellular signals. And so these integrins can be multiple different conformations. They can bind different conformations of extracellular matrix proteins like fibronectin. And when they bind them, it causes some intracellular signaling cascade. And we'll talk about this a little bit more when we talk about the cytoskeleton as to what actually are. But if you think of the cytoskeleton of the cell as a matrix of actin, these integrin binding causes some signal to propagate through the actin network. And these, this word dynamic reciprocity, we can think of this as just the interaction between cells and their matrix. So cells are going to bind the matrix through integrins, and these integrins are going to transmit some signal to the cell, and that is going to change something about the cell, and maybe that cell is then going to change something about the matrix, and then this process starts back over again. So matrix influencing cells, cells influencing matrix. This is the reciprocity piece, so it's a reciprocal relationship, and this is dynamic, so it's always changing depending on the nature of the extracellular matrix and the nature of the cells that are existing in it. Yes. So the protein is going to be more than just that 3-letter sequence, right? So that can be where it binds to, but this is really present in the journal club paper, is that it is going to bind better to some types of proteins than others. So it changes the strength of binding to certain types of fibronectin or to certain types of collagen or other things like that. Does that make sense? The RGD is always there. It usually does bind to the RGD, but also to other sites as well. So we'll talk about in fibronectin, there's like a synergy site, and the binding of both of them together enhances the overall binding of the integrin to fibronectin. Good question. Thanks for clarifying that. Okay, so where these integrins are binding, they cluster together. So multiple dimers of integrins kind of move together. They diffuse laterally on the lipid bilayer, and they create these complexes, and these complexes are known as focal adhesions. So this is a little bit confusing because are where the extracellular matrix is on the bottom and the intracellular side is on the top. You don't, again, have to know any of these adapter proteins right now, but ultimately these complexes of integrins are recruiting a bunch of different proteins on the inside of the cell, and that causes the signaling cascade. This is happening at these focal adhesions. And so we actually visualize this in a cell where here on the image in the bottom, the red is the nucleus. The whole cell structure is not really stained, but you can see these localized sections in yellow are where the focal adhesions are. And so these are the spots where the cell is binding to the matrix. And this is really important for growing cells outside of the body, but also important for the cells to maintain existing normally and healthily in the body. Sorry, I'm kind of flying through this. I'm worried we won't have enough time. But we're going to talk a little bit about cell-cell adhesion receptors. So there's 4 different types that we're going to talk about. They fall into 2 categories, and these are known as homophilic or heterophilic. And so similar to other nomenclature, homo just means the same, hetero means different. So the homophilic associations are when there's 2 receptors on cells that are the same and they recognize each other and bind. Whereas the heterophilic are when 2 different things are binding each other, both on cell membranes. So the first class we'll talk about is the cell adhesion molecules, or CAMs, and we can name these like NCAM, referring to neural cells, VCAM, like vascular cells, ECAM, endothelial cells, EpCAM, epithelial cells, and this is actually composed of a part of an antibody. So this Ig is immunoglobulin, and this is a part of the entire antibody structure that we'll talk about later in the class. Integrins are another type of adhesion molecules, and similarly, so N-cadherin, E-cadherin, and these are another type of homophilic association, but these are just protein structures rather than the immunoglobulins. And these are calcium dependent, so they require calcium in the interstitial space for this. The heterophilic associations, these are going to be lectins. So these are proteins that bind oligosaccharides or glycoproteins. So again, these are these sugar molecules out of cells. And we talked about can also bind cells. So we traditionally think of them as binding the extracellular matrix, but they can also heterophilic association. Yes. The homophilic means that there are two molecules that are the same that bind each other, and the heterophilic means that there are two molecules that are different binding each other. Okay, so we talked a lot about what makes up the extracellular matrix and the different components, but where does it come from? So a lot of different cells can make extracellular matrix. A lot of cells make some amount of extracellular matrix proteins, but fibroblasts are their primary purpose is to synthesize extracellular matrix proteins, and collagen in particular is a main protein that they synthesize. So the structure, they reside in this interstitial space, and this is exactly where they're secreting all of these proteins. And the state of them differs based off of what they produce and how they respond to their environment. So in a quiescent state, they synthesize some amount of extracellular matrix protein or collagen in particular, but not a lot. And then some kind of trigger they can activate. And a lot of this is wound healing, which we'll talk about. And they'll start laying a lot of different types and amounts of collagen in response to injury or inflammation. So we can think of these as different states or different subtypes of fibroblasts. I just like this video. Fibroblasts are the best cell type, and they're really cool. This is just a video of a fibroblast moving within a collagen matrix. So these cells, even though we can think of them as quiescent, which is generally thought of as like a sleeping or a quiet state, they still move around quite a bit in the matrix space. Okay, so coming back to the disease, we're gonna walk through a little bit of difference between a normal wound healing cascade and what goes wrong in fibrosis and what is the case in pulmonary fibrosis as well. So we just have here an epithelial layer, and so if this was the lung, up here would be the air space, and this interstitial layer, which in the lung is really thin, and some kind of injury happens. So maybe that is something happens to those epithelial cells, they get injured, they break down the basal lamina to some coagulation cascade. So there is there's a release of blood that causes fibronectin to bind and activate fibrinogen to fight. This triggers immune cells to come over, such as macrophages, and these will release all sorts of signals into the interstitial layer, such as cytokines. And these cytokines can change the state of fibroblasts into this activated fibroblast. So this is one way that these cells can change states because of the signals from immune cells. And then now we know that there's something going wrong. And so they'll start laying down a bunch of new extracellular matrix. Is to try to heal where that injury has occurred. So the blood clot isn't going to last for a long time. And so this matrix protein can take the place of that. And so once that is stable, then that can trigger other cells to repopulate the basal lamina for the epithelium to divide and continue in a healthy way. And then everything goes back to homeostasis or a normal amount. So the immune cells stop secreting their cytokines, the fibroblasts unactivated. And then eventually the other cells in the environment will remodel or break down that protein that has been secreted, and then it'll be just as healthy again. Does that make sense? I know I walked through it pretty fast. Okay. In fibrosis, what happens is these immune cells are triggering the fibroblasts, but then they just never stop laying down extracellular matrix. So these fibroblasts become what we call constitutively active, which means that they're always active, they never revert. And this could be either because these immune cells or because there's something wrong with the fibroblast that they can't recognize those signals. It could be something about the ECM that they're lying down that is triggering them to keep lying down more ECM. But these fibroblasts are creating all of this extracellular matrix protein, collagen particularly, that is leading to this persistent scarring. So that in the lung, this happens in this interstitial space between the blood vessels and where the alveoli are, these places of gas exchange. And this creates a thickness in this space that is permanent. There's no cells there to degrade it. The cells that are there are contributing to more ECM, and this is leading to progressive scarring. Any questions about this? All right. Well, there's a bunch of professors in the department that do research in this area. Dr. Tom Barker is one of them. Stephen Cagliari is my advisor, and both him and Dr. Don Griffin in the department both come at fibrosis from a biomaterials or hydrogel kind of viewpoint. Okay, yes.
Were you gonna do any things or?
What do you think? We've got about 12 minutes. Oh, we have 8 minutes. Okay. Oh,
Why don't you can go over why don't you go over like the journal club over maybe?
okay. Yeah, okay, that works. Yeah, obviously we're a little short on time. That's okay. We love the discussions and the questions. So if there's anything towards the end of the lecture that was unclear because I was trying to rush through it, feel free to just come to office hours. We'll have tons of time to talk through it. Or you can email us or post on Piazza if there was anything unclear. But we wanted to do a journal club today. We'll just introduce the structure and the layout and a little bit of what to expect. It also gives you a little more time to sit with the material since the paper is on integrins and fibronectin. So a couple of things to think about when doing the journal clubs. These will be group-based activities. So these are really kind of technical concepts and figures. So it helps to divide and conquer, to split up in your group, to look into different things at once and to come together to talk about it. This could be unknown terms, what the y-axis means, what the figure caption is saying, any of these things. I would strongly suggest you use resources available to you. So there's only a few of us on the teaching team, and there are lots of groups that will be working on this simultaneously. So if there's things that you are unsure about, I think trying to look into them first before asking us would be a good place to start. If you're not finding anything helpful in your search, then we would love to help you and just guide you in terms of what things to look into, what things to search for, how to best go about understanding these figures. But really the goal of this exercise is to just get you more confident in learning how to figure out these papers on your own. And in terms of what to focus on when you're going through these these figures, the question you really want to be asking yourself is, what is the purpose of this experiment or piece of data? What are they trying to show here? How does this fit in to the overall structure of the paper and the argument of the paper? And I'll talk a little bit about that in terms of this paper, so that you guys can ask any questions up until next Tuesday. So we're also going to be, another reason why we definitely couldn't do it today, we want all the groups to go around and discuss what they found. So to share with the class what the takeaway from their figure is. So these should be really brief. There's like 10 groups that'll all be working on this. So we don't want like 5 minutes per group because that's almost the whole class. But you just want to say as briefly as you can what the figure was looking at and what the main takeaway is, and maybe some kind of principle about the method if you get to that or if you have time. So the paper that we'll be discussing is actually a paper published by the lab of Dr. Tom Barker. And this is a paper back from 2017. So it's not a new paper, but it's really interesting and I think relevant to this class. So it's called The Detection of an Integrin Binding Mechanoswitch within Fibronectin During Tissue Formation and Fibrosis. So it's also the disease application of this paper is fibrosis, and it focuses on this binding between integrins and fibronectin. So this word mechanoswitch is really important here, and this is just referring to is pulled on, it changes conformations. And what they're trying to show here is this hypothesis is that if you pull on fibronectin, it changes conformations. So first, how to prove that? And then second, the hypothesis is that extended conformation will bind different types of integrins. And so from that, in terms of its implications or its relevance to this disease of fibrosis, the takeaway that they're trying to show here is that these integrins that are binding different conformations of fibronectin preferentially are going to be more or less in fibrosis. So a lot of aspects of this paper are going on. This is a really difficult thing to prove and to probe specifically in the lab. So there's two main types that are utilized. We'll go over this quickly, but a main one is protein engineering. So you can make proteins in the lab that model proteins in the body, and what they really wanted to do here is to make certain types of the fibronectin protein that are kind of locked or frozen in these different conformations that they're trying to study. So either in a more tightly packed state or in an extended state. So you'll see a lot of different protein figures that are based on what are these different conformations that they make with these new engineered proteins in the lab. And then another main thrust and tool used in this paper is antibody engineering. So I'm going to talk briefly about the concept behind immunostaining. So immuno just means immune-based, and this is specifically antibodies. So antibodies are these proteins that your cells produce that can bind specific things, and we can use them in the lab to probe things to see if that's there. So you can use an antibody and you can put it on your sample, and it's designed to bind a certain thing. And so you can take that antibody that binds a certain thing and you can attach a fluorescent molecule to it. And so then if you shine a fluorescent laser, you can see where that antibody is. So for this paper, what they did is they made an antibody that would bind to a specific conformation of fibronectin in order to visualize that conformation, not just in the proteins that they engineer, but in real cells and tissues. So I'll just go ahead and end it there. If you have any questions, feel free to ask office hours, Piazza, email us. But have a good rest of your day.